వచ్చింది.
Good morning, students. This is G.RAMA JL Botany, and welcome to webinar 4 session. Today, we are going to discuss C4 pathway. This pathway is also known as Hatch-Slack cycle by the name of the scientist who gave the complete cycle information. And this Hatch-Slack cycle is seen in C4 plants like maize, sorghum, sugarcane, and euphorbia like plants. And these C4 plants, these are by showing some different adaptations or by showing some structural changes, they escapes from the photorespiration. So it's a cycle which adapts adaptations to avoid photorespiration. And this C4 plants, how they adapt themselves. In C4 cycle, actually a cycle which is evolved to avoid photorespiratory loss. So all these C4 cycle showing plants, they show some adaptations to avoid photorespiratory loss. Then in our previous chapters, we discussed about the photorespiration problems. And this problem is because of a changing behavior of a Rubisco enzyme. And this Rubisco that changes its nature according to the environmental conditions like high temperature and high oxygen concentration. Under these two extreme conditions, this Rubisco that acts as carboxylase or oxygenase. So this changing behavior also helps or supports this photorespiration, and which is a problem or which is a hurdle for the normal plants. And to escape from this problem, C4 plants, they show some adaptations. Anatomically, they show some different characters. Until and unless the plant shows some adaptations, they suffers from extreme conditions like high temperature and high oxygen concentration. So by this, we can understand that C4 plants are normal plants only, but by showing some adaptations, they escapes from the photorespiration. And this photorespiration is a problem creating process in plants because of a changing behavior of rubisco. And this rubisco changes its nature under the extreme conditions like high oxygen concentration and high temperature. Okay, then next. Just only we said that C4 plants show some anatomical differences from the normal leaves. And those anatomical adaptations by C4 plants, what are they? Just those are discussed in adaptations in C4 plants to avoid photosynthesis. Under this statement, we can discuss two important points. They are, number one, because of having dimorphic chloroplasts. Di means two, morphic means different forms. So usually in normal plants, chloroplasts are of only one type, and their structure, we know it very well, that even though, one second, that the normal structure of a chloroplast, if we observe it, it is a double membrane bonded structures, ovoid structures. Inside these membranes, some gelatinous substance, which is called as stroma. Stroma will be there, and in that stroma, stacks of thylakoids, which are called as grana. Granal stacks are arranged randomly, and this granal stacks are interconnected with one another by means of some thread-like structures or some strands, which are called as stroma, thylakoid strands. And these strands, stroma lamellae, they are called as stroma lamellae, and this helps in binding the granal stacks. This is the normal structure of a chloroplast. And the C4 plants, they exhibit some little bit different symptoms or some characters. So based on that, C4 plants, they develop two types of chloroplasts. Number one is Grana type of chloroplast, and the second is Grana type of chloroplast. Grana type of chloroplast. This chloroplast, they would have grana in the chloroplast. That means this chloroplast that consists of thylakoids, and thylakoids are would be arranged in stacks, which are called as grana. And this grana, granas are connected by means of stroma lamellae. That is, grana type of chloroplast is similar to that of normal chloroplast. And this type of chloroplast, that means a granal type of chloroplast, they are present in, or they are found in mesophyll cells. That means in leaf anatomy, upper and lower epidermis, or in between the upper and lower epidermis, there is a tissue which is called as a mesophyll tissue, and this mesophyll again that is classified into palisade and spongy tissue. So this mesophyll tissue that exhibits, or in mesophyll cells, we can find this granal type of chloroplast, and this granal type, they are similar to that of normal chloroplast. And in thylakoids, in normal chloroplasts also, thylakoids, they helps in storing pigments like chlorophyll or others. So these pigments are the sites for performing light reaction. So in granal type of chloroplast, there is a specificity which is similar to that of normal chloroplast by having a double membrane, and inside the stroma, and in stroma granal stacks which are connected by stromal lamellae. And in each and every thylakoid, pigments are preserved or pigments are present, which helps in performing the light reaction like non-cyclic cyclic photophosphorylations, through which assimilatory power will be synthesized. So this is about the granule type of chloroplasts, and these are found in mesophyll cells of the leaf of C4 plants. Then another type of chloroplasts are agranule type of chloroplasts. And this agranule type of chloroplasts are found in bundle sheath cells of the leaf, not in mesophyll cells. They are found in bundle sheath cells. And these bundle sheath cells are the cells which are bounded around the vascular tissue. That means around the xylem and phloem tissue, a single cell thickness membrane is there. That is called as bundle sheath. And each cell, in each cell structure, we can found this type of, that means agranule type of chloroplasts are visible in this bundle sheath cells. And if we observe the structure of this chloroplast, this chlorophyll would not have grana. Difference between grana type and agrana type is, number one, main difference is in agranal type, this chloroplast would not have grana. They would have only stroma lamellae. That means the granule stacks are absent in agranal type. And because of the lack of this granule stacks, pigments are not available in this type of chloroplast. So if the pigments are absent, then they are not able to perform light reaction. Instead of light reaction, by having this stroma lamellae or stroma, this agranule chloroplasts are very suitable sites for performing C3 cycle. Okay, so this is about the agranule type of chloroplast. So this type of chloroplast would not have grana, and they would have only stroma lamellae, and they are found in bundle sheath cells. And this agranule type of chloroplasts are suitable sites for C3 cycle. So this is about the dimorphic chloroplasts. That means chloroplasts are showing a difference in their structure. As a result of this deviation, they are performing different activities. Grana type is performing, or grana type of chloroplasts are the sites for performing light reaction, especially for non-cyclic photophosphorylation. And agranule type is the site for C3 cycle, which are found in bundle sheath cells. That's it. The next, second type of adaptation by the C4 plants is Cranch type of anatomy. What is the meaning of Cranch type of anatomy? That means C4 plant shows Cranch type of anatomy. That means cells of bundle sheaths are arranged in a wreath-like manner. You know the meaning of wreath. Wreath means in a single circle manner. That means bundle sheath cells, they are arranged one after the other in one cell thickness around the vascular tissue. That is called as nothing but wreath-like manner. So in bundle sheath cells, cells are arranged in a wreath-like manner. That means cells are arranged in a circular shape. And in this bundle sheath cells, we are getting agranule type of vascular bundles. That means a C3 cycle will be performed in bundle sheath cells. The next, here, look at the diagrams. Here, two types of two types of plastids are there, chloroplasts are there. See here, MCP, that means a mesophyll chloroplasts. And here BSCP, that means bundle sheath chloroplasts. See, in this mesophyll chloroplasts, granule stacks are there. They are appearing in dark color. And next, these granule stacks are interconnected with one another, one another by means of stroma lamellae. So these light color structures are stroma lamellae, and these dark color structures are nothing but grana. And here in this diagram, you can see, so this is a grana type of a chloroplasts which are found in mesophyll cells. And here it is agranule type of a chloroplast which are found in bundle sheath cells. See here, these light colors strand-like structures, these are stroma lamellae. And we are not observing any dark color places which are found in mesophyll cells. See, these dark colors structures are nothing but grana, isn't it? And here in agranule type, we are not seeing any dark color structures. That means grana is absent, or granas are absent in agranule type. Only stroma lamellae are there. But whereas in mesophyll cells, grana type of a chloroplasts are found. So that is the difference between the grana and agrana. The next, cranch type of anatomy. See here. This is the diagram of C4 plants leaf anatomy, in which you can very clearly observe the cranch type of anatomy. See here, the centrally located cells are vascular tissue, and which is bounded by a single cell thickness cells called as bundle sheath with chloroplasts. And this bundle sheath chloroplasts are of agranule type. And mesophyll cells around to this bundle sheath, external to this bundle sheath cells, we are observing a light color structure cells which are having so many chloroplasts, which are of granule type of a chloroplast in mesophyll cells. So this cranch type of anatomy means cells, large cells in single cell thickness, and one after the other in a single layer, cells are arranged in a wreath-like manner or in a circular manner, in which you will find only a granule type of chloroplasts. So that is about the cranch type of anatomy. So C4 plants shows cranch type of anatomy. That means cells of bundle sheaths are arranged in a wreath-like manner or in a single circular manner. In this diagram also you can observe the structure of the C4 plant's leaf. Mesophyll cells in which chloroplasts are abundant. They are of grana type. And here in the center of this leaf's anatomy, this is a bundle sheath cell. This bundle sheath cells, they are arranged one after the other in a single cell thickness. And these structures are, these are the cells of bundle sheath, that special type of chloroplasts here. So those are agranule type. So this is about the cranch type of anatomy in C4 plants. In the next slide, what is the use, or by this adaptation, is there any benefit for this leaf, C4 plants leaf? That is under the caption, use of cranch type of anatomy. What is the use of cranch type of anatomy? See, what is the advantage of this anatomy? By having this type of anatomy, is there any use for this C4 plants? Yes, granule chloroplast would be in mesophyll cells, and agranule chloroplasts would be in the bundle sheath. That was discussed in the previous slides only. So, granule chloroplasts very clearly we must remember this point, that granule type of chloroplasts are found in only mesophyll cells, and the agranule type of chloroplasts are they would be in the bundle sheath cells. And one thing, wherever there is granule chloroplasts and thylakoids, then definitely that is the place for site of light reaction. That means this granule type of chloroplast, definitely they helps in receiving the sunlight from the epidermal, upper epidermal cells, and they traps the sunlight, and they helps in running the light reaction changes. So this granule type of chloroplast, wherever they are, there is the site for light reaction. Then next, wherever agranule chloroplasts are there, there is a stroma, and their C3 cycle definitely takes place. Just now only we discussed it in our previous slides. So having a granule type of chloroplast in mesophyll cells definitely indicates a site of a light reaction, and wherever agranule chloroplasts are there, they are the definite sites for C3 cycles. So this is one important advantage. The next, what happens by this type of different chloroplasts in different parts? That means in mesophyll cells, we are observing granule type of chloroplast, and in bundle sheath, we are observing agranule type of chloroplast. Then why it is? Why the granules are only restricted to mesophyll, and why the agranule are restricted to bundle sheaths only? Why don't vice versa, or why don't in reciprocal? Means there is a reason for this type of arrangement. And what is that? Let us. From upper surface cells of the leaf receives sunlight and passes it into the mesophyll cells, where the light reaction takes place. As a result of non-cyclic photophosphorylation, photo is released as a byproduct. Yes, reason for this type of arrangement, that means a granule type in mesophyll and a granule in bundle sheath. There is a specific reason, and that reason is in Grana, that means in a Grana type of chloroplast, a full-fledged organelles are there, which are thylakoids, in thylakoids pigment. And this pigment is most essential factor in receiving the sunlight from the upper epidermal cells. And these thylakoids are absent in agranule type of chloroplasts. That's why Grana type of chloroplasts are situated in mesophyll cells. That is the advantage. By having this Grana, by having this thylakoid, and by having these pigments in thylakoid, they helps in trapping the sunlight through the upper epidermal cells. So that is the advantage of this having Grana type of chloroplast and mesophyll. So by trapping the sunlight, by trapping the sunlight energy, this light is absorbed by the pigments, and they helps in running the light reaction. And during this light reaction, we have already discussed that light reaction will be completed in two stages. One is cyclic and the other one is non-cyclic. And in cyclic photophosphorylation, ATP will be synthesized by transferring or by exciting two electrons from photosystem one, and by receiving or by traveling through various electron transporters, again it reaches, or again it comes back to the same photosystem. By synthesizing this ATP molecules, it reaches the same pigment system. That's why it is called as cyclic photophosphorylation. Cyclic means in a circular way, and photo means in the presence of light, and phosphorylation means synthesis of ATP. That means that ATP synthesis is taking place in presence of sunlight, and the changes are going in a circular manner. That's why it is called as cyclic photophosphorylation. And the second is non-cyclic photophosphorylation, where two types of systems are involved: pigment system 2 and pigment system 1. First, reactions starts in pigment system 2. That means by receiving the photons by the pigment and the accessory pigments. This accessory pigments also transfers the photons to the reaction center. And by the excess of these photons, it gets excited and releases electrons. And these electrons are accepted by primary electron acceptor. And from high energy gradient to low energy gradient, these electrons are transferred from high energy gradient to low energy gradient through various electron acceptors. And finally, electrons are accepted by pigment system 1, but not they comes back to the pigment system 2 or photosystem 2. It means released electrons are transferring through various electron acceptors, and finally they are reaching to the pigment system 1, but never they comes back to the same pigment system 2. So while this movement or while this traveling of electrons from different electron acceptors to photosystem 1, they synthesizes ATP molecules. And here one problem is created, that is loss of electrons in pigment system 2, and how that will be compensated, or how the electron loss will be replaced. Means by means of photolysis. This photolysis of water means what? That means in the presence of light, one H2O molecule is dissociated or splits into H plus and OH minus. And furtherly, this OH minus again splits into oxygen and protons. Electrons. And these electrons are shifted to pigment system 1 to replace the electron loss. And this oxygen that comes out as a byproduct. That's why during photosynthesis, oxygen is released. We are showing it in the equation, isn't it? So this photolysis of water helps in replacing the electron loss in pigment system 2. So oxygen is coming out as a byproduct. And then what about the protons? This protons helps in reducing the NADP plus into NADPH plus H plus. So for reducing this NADP plus, these protons are useful, and electrons are, whatever electrons are released through this photolysis helps in compensating the or fulfilling the electron loss in PS2. So why we are discussing all this is, here what we are saying in granule type of chloroplast, through, that means a light reaction takes place in granule type of chloroplast, and in this light reaction through non-cyclic photophosphorylation, O2 is released as a byproduct. So that is the importance of having granule type of chloroplast in mesophyll. Then next, what about the agranule type? Why the C4 plants adapted dimorphic chloroplast means because of these reasons. First of all, to perform non-cyclic and or to perform light reaction in which through non-cyclic photophosphorylation to release oxygen into the atmosphere. That is one reason, by having a Grana type of chloroplast. Then by having a Grana, what is the adaptation? What is the use to the C4 plants? See, let us discuss about that. By having this agranal type of chloroplast in the in the bundle sheath cells. By having this, usually in normal plants also C3 cycle that will be performed in stroma. So C3 cycle, which is mainly it is a Rubisco dependent. That means it depends on the Rubisco enzyme. And here this C3 cycle in C4 plants, C3 cycle that is shifted into the deeper layers cells of bundle sheath, where temperature is less and oxygen concentration is also less than the upper mesophyll cells. So the C3 cycle, site of the C3 cycle is in the cells of bundle sheath, where temperature is less and this concentration of oxygen is also be less than the mesophyll cells. Why? Why means these mesophyll cells are very closer to the upper epidermal cells, and they helps in transferring the temperature or photons from the atmosphere into the mesophyll cells, which are closer to the upper epidermal cells. And as a result of that, temperature is high in mesophyll than the lower tissues. And oxygen concentration is also high. Why? Because through non-cyclic, it is coming out as a byproduct. So O2 concentration is also somewhat higher than the lower tissues. That means that in mesophyll cells, temperature and oxygen concentration both are high. And this high temperature and high O2 concentration, they changes the nature of Rubisco enzyme. And this Rubisco enzyme is essential for running this C3 cycle. C3 cycle is Rubisco dependent. That means Rubisco RUBP carboxylase helps in starting the C3 cycle. Under the less temperature and less oxygen concentration only, the RUBP acts as carboxylase. But in mesophyll cells of C4 plants, there is high concentration of temperature, temperature and high oxygen concentration. And under those conditions, RUBP cannot act as carboxylase. It acts as oxygenase, which is a problem for C3 cycle. So to avoid this situation, this C3 cycle is shifted into the bundle sheath cells, where oxygen concentration is less than the mesophyll, temperature is less than the mesophyll cells. That's why this is the second use of a shifting C3 cycle from mesophyll cells into the bundle sheath cells. Then next, about the Rubisco enzyme. Rubisco acts as oxygenase, where the temperature and oxygen concentrations are very low than the other parts. Rubisco acts as oxygenase when the temperature is high. So C4 plants have changed the site of C3 into bundle sheath cells, where the temperature and the oxygen concentration is less than the mesophyll and uppermost cells. So this is the reason for changing or for shifting the C3 cycle from mesophyll into the bundle sheath cells. Then the temperature, about temperature, now it is cleared. That means in mesophyll cells, the temperature is high. In mesophyll cells, temperature is high because of the uppermost cells transfer of heat from outside into the mesophyll cells. Then next, that was solved by that temperature effect is solved by shifting the C3 cycle into bundle sheet cells. Then second reason or second problem, that is about the oxygen concentration. This oxygen is released because of non-cyclic photophosphorylation that takes place in the thylakoid membrane of granule chloroplasts, which are present in mesophyll. That means in mesophyll cells, by performing this non-cyclic photophosphorylation, oxygen concentration gradually increases. And here O2 is released or O2 is released as a result of non-cyclic. As a result of that, O2 concentration gradually increases. In bundle sheet cells, there are agranule type of chloroplasts, and in which a non-cyclic photophosphorylation could not be performed. As a result of that, O2 will not be released. That means O2 concentration will be less than the mesophyll cells. So in bundle sheet cells, O2 and temperature both are less. This extreme conditions are under the control where the Rubisco oxygen is taken care of. So under these conditions, Rubisco acts as oxygenase. So both the problems are solved. That means a high temperature and high O2 concentrations. They changes the nature of Rubisco. That's why in mesophyll, these two conditions are extreme. That's why there C3 to perform the C3 cycles, conditions are not favorable. That's why it is shifted to bundle sheet cells where the temperature and oxygen concentrations are less. In C4 plants, adaptations by having this dimorphic chloroplast and a cranch type of anatomy, they are escaping from the photorespiration problem. That's clear. Next, process of C4 cycle. In C4 cycle, it will be completed in two different parts. Number one is mesophyll cells. Number two is bundle sheet cells. In mesophyll cells, just now only discussed that mesophyll cells are having a granule type of chloroplast, which helps in performing light reaction. And bundle sheet cells, they are helpful in performing C3 cycle by having a granule type of chloroplast. Let us discuss about the individual changes in mesophyll and bundle sheet cells. First, we'll discuss about the mesophyll. In mesophyll cells, which are very closer to the upper epidermal cells, and in upper epidermis, here and there stomata are arranged, and this stomata helps in the diffusion of CO2 from the atmosphere into the mesophyll cells. So CO2 enters into the mesophyll cells through the upper epidermal cells, and that reacts with H2O in presence of enzyme like carbonic anhydrase. First, CO2 that combines with one water molecule, and in the presence of one enzyme that is carbonic anhydrase. It results in the formation of carbonic acid. Okay, so CO2 reacts with H2O molecule in the presence of carbonic anhydrase and results in the formation of carbonic acid. The next, carbonic acid, it dissociates HCO3, that dissociates into H plus and HCO3, that means bicarbonate. And this bicarbonate is accepted by PEP, that is phosphoenol pyruvic acid, which is a 3C compound. And this